Hello, and thank you for joining us today for a Science Series webinar with Imugene CEO and Managing Director Leslie Chong. We are also joined today by a very special guest, Dr. Yuman Fong. By way of introduction, Dr. Fong is a pioneer both in the operating room and in the laboratory. He does robotic surgery, he designs robots, and creates viruses. He's an internationally recognized expert in the cancers of the liver, pancreas, gallbladder, and bile ducts. He's led the research effort to employ genetically modified viruses to destroy cancer cells and is the inventor of Imugene CF33. And here's a little factoid. The F, I've heard, in CF33 is actually for Fong. Dr. Fong joined City of Hope in 2014 after more than 2 decades at the renowned Memorial Sloan Kettering Cancer Center in New York City. He's both author and innovator. He's developed many new surgical techniques and instruments and has written and edited hundreds of scholarly articles, as well as nearly a dozen textbooks. I also heard that Dr. Fong wrote comic books about the immune system being the superhero against cancer. So you're clearly a man of many interests and many talents, Dr. Fong. So thank you for taking time to be here today. Before we dive into the specifics, I'd like to kick it off by learning a little bit more about you. Why is it that you do what you do, and what drives you to invent?
Well, a lot of folks say that I go do many different things, but I really only do one thing. I'm trying to treat patients with cancer. Whether it is that I am doing robotic surgery or open surgery or designing cancer therapy such as viruses and cells, it's all about treating the patient. And it's all about trying to defeat cancer. And so I keep saying, again, I chant to my patients that what we're really trying to do is more cures, less invasive. And I just couldn't think of a better way to do non-invasive therapies than to send in viruses that will go and hunt down cancers and kill them. And that's what the whole idea behind oncolytic viruses are.
I know that you and Imugene have collaborated for a few years now. Why did you choose to work with Imugene?
Well, it's been quite a collaboration. So in 2014, I came out to City of Hope, which is in a suburb of Los Angeles. It's a dedicated cancer center, and this is a hospital, a research hospital that's entire mission is to take care of patients with cancer and to find new therapies for cancer. I came out here because the City of Hope actually had a entire complex of laboratories that could make clinical grade medicine, meaning that whatever I design in the laboratory can become human medicine in a very short time. I was working very hard in New York at Sloan Kettering to try to make new cancer therapies, but it was taking too long to go to humans and to go and make impact on cancer. So I came out in 2014 because I thought it would be a shorter path. We designed a whole series of new viruses that are enormously promising. And it was at that time, in about 2018, 2019, that Paul Hopper and Leslie Chong knocked on my door and said they were in the midst of trying to find new immunotherapies for cancer and thought that the things that we had created were promising and wanted to work together. And after a few conversations, I was convinced that they were committed. They
Mm.
had the resources to go ahead. They had the expertise to go ahead. And the partnership has proven to be wonderful because not only have they really been very focused on going to humans, testing things and finding out what works and what doesn't work, and keep moving forward in a positive direction towards trying to defeat cancer. But the partnership's been wonderful in the sense that they've always fed back to me the results. And then we could actually go and redesign and push forward trials that make sense and are making huge progress. So again, this partnership started in 2020. It's only been 4 years, and to be in multiple trials, to have many patients that have had responses to therapy, and then to really have a path forward that actually can make all of this become real human medicine. in such a short time. It's been a wonderful relationship, Heather, and I look forward to the immediate and the future of trying to make all of this human medicine.
That's wonderful. Leslie, did you want to chime in at all from your perspective on how the partnership came about?
Well, so it's exactly as Dr. Fong said. We met and we pretty much got along incredibly well right away. We knew that both of us wanted to develop medicine for humans. Both of us were very much into getting CF33 into patients ASAP, and that's exactly what we've done. And my team, along with myself, when we hear any good news, all of us tend to call up Yuman to say, Yuman, I think we might have a response. And so that the first response that we got, I know it was a nail biter, but we immediately called, a few of us actually had called Yuman to say, your virus is working and it's working in these kind of indications. So we keep building on our relationship as Dr. Fong says, and we hope to benefit a lot more patients moving forward.
That's wonderful. Moving on to oncolytic viruses in general, Dr. Fong, can you help explain to us how is it that oncolytic viruses specifically target and kill only cancer cells, but they spare healthy cells?
Well, it's come from so this idea that there are viruses that could actually target, infect, and kill cancer cells. That idea has been around a long time, Heather. Ever since the 1950s and 60s, there have been many reports of patients getting natural virus infections and having suddenly a remission in cancer. And back then, the investigators had only had natural viruses to go on. So there were investigators around the country and around the world that were taking natural viruses, viruses like measles viruses and polio viruses and West Nile virus, and giving it to human beings to see what would happen. And they did see some cancers regress and some many patients treated. But natural viruses are just too toxic. And so starting in the 1980s and 90s, we started designing these because starting in the early 80s and 90s, we started having tools to be able to do genetic engineering of viruses to make them much more specific for cancer. But this whole idea of finding cancers or finding viruses, this whole idea of finding viruses or designing viruses that only infect cancer, not far-fetched, because in nature, that's what viruses do. They like only infecting certain cells. So hepatitis viruses only infect liver cells. Meningitis virus only infects neural and brain cells. And so this is the whole idea is in this field is finding viruses and designing viruses that will go and only infect cancers. And we think we've come close and probably have done it with CF33. This is a virus where we created many, many viruses that potentially can infect only cancer and tested them against hundreds of tumors that tumor cell lines that and cared about only the viruses that infected all of them. Okay. And so unlike the last generation in the 80s and 90s, we were trying to find viruses that would only infect skin cancer or only infect breast cancer or colon cancer. This time around, I said, let's go find viruses that only infect cancer, but all cancers. That way, we can actually design a medicine that affects not hundreds of thousands of people or a few million people, but all potential cancer patients. And so this is how CF33 was born in the laboratory. And so far in testing, not only have we been finding that it's been very safe, it's been remarkable that we've seen biologic activity in many different types of cancer.
Actually, this is a great segue to Leslie to talk about the programs, CF33 vaccinia programs. I know that the MASS trial is ongoing. And then if you could also talk about Oncolytics as well. And I know that you've had recent announcements go out. So if you could just give a status update on the programs.
So as Dr. Fong alluded to, we have had a lot of safety, and so we're able to dose escalate. And I just want to just brief everyone on what it means to find a dose to move forward into phase 2 or phase 3, or for something to become a drug. Phase 1 is all about finding that nice triangulation between response rates, where you are affecting the tumor and hopefully shrinking the tumor, and then also toxicity, balancing that level of toxicity, and then also sustainability. So that's why phase 1s are dose escalating. So you dose escalate. Sometimes you stay at the same dose because you're looking for something else, a meaningful signal. And then it's also wonderful to find a pan study like this for an indication. So an indication such as bile tract cancer, which is, we find it incredibly lucky, and knowing that we're causing a level of efficacy in these kind of patients, because it's an unmet need, meaning there's not a lot of drugs out there for these patients. So the FDA tends to be a bit kinder when it comes to a registrational path, meaning to market this drug. So we're quite exuberant about the fact that we're in bile tract cancer or cholangio cancer. It is right in humans were, wheelhouse, which is incredible, because I don't think that he thought that we would go after cholangio. I think we thought that we would be in lung or, head and neck or something. So to find a response rate in an unmet need, we already have fast track designation from the FDA to go fast. And Imugene is all about going as fast as we can. But during the phase 1, you really want to find that dose, because you can't backtrack. So the indication that we are looking at now is bile tract cancer. We will go fast in this particular expansion trial. If we find a level of tumor shrinkage across the 10 patients, then we will turn that into a fast acting phase 2. Hopefully that will go fast. And then hopefully CF33 could actually be a drug.
Dr. Fong, Leslie mentioned bile tract cancer. Were you surprised that it worked?
I was
Or that
not
we've seen-
surprised it works, but how well it worked, I've been very impressed. So let me just reiterate some of the things that Leslie has said. Okay, we are right now in the midst of three different parts of developing CF33. One is that we're still in the midst of going higher doses in all kinds of cancers. Okay, that's the mass trial, where we take patients with cancer of any type, and we're willing to put them on trial and give them medicine to see whether we can actually go and impact and shrink tumor and prolong lives of these patients. Second is that we've had such good response, meaning that we've seen a patient with this bile duct cancer, which usually is completely resistant to chemotherapy and radiation. And in one of these patients, the tumor completely disappeared. And now, months later, without any therapy, the patient is still without cancer. And another one where the disease just sort of froze, where did not progress anymore. And those were the first two we treated. And this is in a disease where we have not had many solutions at all. And almost everybody who has metastatic bile duct cancer dies. And so we're quite excited, and the FDA has given us fast track to move ahead with a bigger trial to prove that in this cancer, our CF33 virus is useful and should be a medicine. But the third part of where we're going is even more exciting. And in all of these things that we're doing, we've come to realize that when we give virus, it's not only a direct killing of cancer that the virus does. The virus also stimulates the immune system to recognize the tumor as something that's foreign, and for immune cells to come and try to attack it just from an immune basis, because our human immune system is quite sophisticated, and it's really good at killing all kinds of things, including bacteria and viruses and cancers, if it could recognize whatever it is as foreign. And our virus is now making the body recognize that the tumor is actually something foreign that should be attacked. And then we're taking the next step beyond that, and that's what Oncolytic is that beyond just non-specific stimulation of the immune system to recognize the cancer, we're now tagging the cancer with a protein that doesn't usually exist in that tumor, and then giving all these therapies that are targeting this very useful target called CD19 that has now become a useful target for, and curing lymphoma and leukemia. But now we're using this tag to sort of tag any cancer to allow for all those drugs that have been designed for leukemia and lymphoma to come and complete the killing of cancer. And from that standpoint, if you think a little bit about how we now fire smart bombs at the enemy in war, okay, where we go and laser target the tumor, the target, so that the missiles and the bombs know where to go. That's what we're doing, okay? And yeah, not only is our virus going in and starting the killing of the cancer, it's now tagging the tumor so that these very specialized drugs and immune cells that are targeting CD19 could come and complete the work. And so that's our Oncolytics program, and that is underway. And we are just very excited because in all three parts of this program, we're now seeing results. We're seeing positive results, and we're seeing encouraging results, and that now lead us in the path forward, hopefully, to really a series of medicines for humans.
And Leslie, I know that Imugene announced recent news related to Oncolytics. Do you want to give a status update on where we are?
Absolutely. So Oncolytics is CF33 that covers a solid tumor with a CD19 flag. So it's just as a guided missile going after a CD19, as Professor Fong had explained, that's exactly what's happening. And we have already dosed our first patient with Oncolytics as well as a CD19 therapeutics in that combination. So it's a beginning to a start of Oncolytic study. We also had announced that Vaccinia has already dosed our first bile tract cancer patient, and as well as reach one of the higher doses in our study.
That's wonderful to hear. So as we're wrapping it up, Leslie, can you tell us what is it that gives you confidence in Imugene and its cancer treatments?
I've possibly ran over 200 phase 1 studies in my lifetime at my days at GSK, Genentech, Roche. And in my phase 1 experience, you rarely get a response. If you do get a response, that's great. That may become a drug. But for CF33 in our first phase 1 study, to see a level of response and stability of disease, be it we freeze the disease or we have led that bile tract cancer patient to live without disease in a very rare, hard to treat population, that's what is so exciting. And the fact that we have Oncolytics, which is novel, brand new, there's nothing like it in this world, to create a drug that may help out so many different solid tumor sufferers that don't have a drug for it. It could be a paradigm shift in how we can treat certain kinds of disease. So yes, I'm quite excited about all the activity that's going on at Imugene.
And Dr. Fong, similarly, what gives you confidence in the future of new cancer treatments to help save patients?
I've just been very, very impressed that the studies that we did in animals that were so encouraging, the results are now being repeated in humans. Okay, and there's nothing like seeing that your experiments that you did in the laboratory actually has relevance in human beings and that the biology is the same. Then the other parts of the confidence comes from the Imugene team. I mean, it's I knew it would be great working with them, but I didn't. I totally underestimated how fast they can move, and how well they can go through these trials and do the right thing and gather the appropriate data for us to keep moving forward. And so again, it's been a joy working with them, but it's also been really encouraging seeing the same results that we saw in animals starting to be seen in humans.
Well, Leslie and Dr. Fong, thank you so much for joining us today and sharing your insights. And thank you to everybody who's watching this as well. Have a great day.